All right, welcome to the middle of March. It's Pi Day, 3.14. It's just a random number with no repeats. So I think it goes on to infinity. There's even a contest for how many numbers you can memorize. I looked it up once, and it's way up there, like tens of thousands of numbers. Could you memorize 10 numbers in a row? So imagine doing 1,000 or 10,000. So there's people that have amazing memories. And one summer, well, a few times you run into, how do they do it? There's different techniques. One really interesting one is imagery. And I tried it once with like a random collection of about 20 different items, and you have to say them in order. And if you learn them today, could you do it again next week? These guys, they can do that with imagery. So, for example, like when you walk in this room, they'll place a few items on table number one, maybe three items. Then they'll come over here and place an item. And so they just visually place things in certain spot. Another technique was in your car. When you reach for the door handle, you put something there, then you get in, you put things on the dashboard. So really cool memory techniques, and that's um, not just a photographic memory, but a way to train your brain to do things. So just interesting thing about pi. Um, and we are going to be doing DNA analysis, as I mentioned. So before we get to that assignment, I'm going to go to our textbook, and I'm going to have you go to the textbook too. We're going to read a few paragraphs together, and maybe I'll pick on some of the 6 people that came in tardy and have you guys read first. So, Andy, you were the first one that came in tardy.
Do I read the?
Right here, DNA collection and comparison. You can read it up on my screen or follow along with yours. I'm in the DNA fingerprinting chapter of our textbook.
It is the responsibility of the forensic investigation team to gather samples from the crime scene or from suspects and then analyze it for a set of specific DNA regions or markers. A match of one marker is not usually unique, but if a sample matches 4 or 5 markers, there is a very good chance it is a match.
So keep in mind that we don't do DNA analysis on genes. We have about 22,000 genes on our 46 chromosomes, but DNA like this also is non-coding. So I don't know what the percentage is. My guess is about 60 to 75% of our DNA does not code for genes, and it's just kind of random spaces. It's interesting though that we have genes that code for proteins, but then we have markers that tell where the genes are, and also there are other regions that help it out. So to control when genes get turned on and off are part of these regions. So you mentioned if you look at one marker, it's not going to be unique. But if they can find 4 or 5 markers that are different between 1 or 2 people, now you're starting to get into the highest probability that you are finding a unique person. Fingerprinting is very unique among people. They say no 2 people have ever had the same fingerprint when you're dealing even with like 100 different differences. With DNA, they only look at up to 13 different regions. So our DNA, as large as it is, they only need to find a few pieces that differ between people because we have so many differences in the non-genes. So keep that in mind. 13 places on our DNA that they look at when they do DNA analysis. Who do you want to read next? You can popcorn somebody's name.
Maggie.
DNA is collected at crimes.
Dueling Maddie. You
Yeah, Maggie.
over here. Okay.
Sorry.
What?
You. Yes. Yeah.
DNA collection?
DNA is collected at crime scenes in a variety of ways using tools such as smears, scalpels, tweezers, scissors, sterile cloths, squares, UV light, luminol, or varietal collection kits for sample collection of suspects or living victims. DNA samples can be collected from saliva, blood, hair strands, skin, finger and toenails, and more liquid tooth with root material.
So in order to get DNA, you have to have a cell. They're going to get the DNA out of the nucleus. There's one other place we'll talk about. So why is there DNA in saliva? Isn't that just like a watery liquid with enzymes in it? Do you guys do this lab in biology class? Take a toothpick and scrape the inside of your cheek and then smear it on a slide? And then you looked at it and there were cells in there? So the cells actually come from the inner lining of your cheek. So every time that you, if you were to spit or lick an envelope, not only do you have the moisture just from water and the enzymes, but invariably you're going to have the cells lining your cheeks called your epithelial cells. So just like we slough off cells all the time, dead skin cells, we're also sloughing them off of our mouth and our intestines. So you have to have a cell. So fingernail scrapings, maybe if they were scratching someone, now they have the skin cells underneath the fingernails. So they'll almost always do the scrapings like that when they're doing an autopsy if it is suspicious. So we have Maggie, Maddie. Who's next?
Catherine.
When blood is collected from clothing, the forensic technician will submit all the clothing that is tainted with blood, or if needed, they will send a small sample to the lab while the crime scene is still being investigated. The technician will also use a sterile cloth square and a small amount of distilled water to absorb blood from the victim or suspect for analysis. When blood is found on furniture, the whole piece will be sent to the lab if possible. If not, then samples will be taken from the furniture. When blood is found on a wall, tub, or some other object too big or too difficult to take to the lab, the technician will scrape the blood into a sterile container for further analysis.
So they can get DNA out of blood because blood contains several different types of cells. It's interesting to note that red blood cells lose their nucleus though as they're mature. If they're immature red blood cells that start in the bone marrow, then those could be used. But when they're mature, they need more space to carry oxygen with hemoglobin, so the nucleus disintegrates. But there's other types of cells in our blood, and that's where they're going to extract that DNA. All right, who's next, Catherine?
Brittany.
When preparing the analysis of DNA for identification of an individual, the DNA is converted into a series of bands called DNA typing. It is important to note that only a small percentage of DNA differs from one person to the next. Therefore, only segments of one's DNA are used to create a DNA profile. There are many different methods that can be used. It all depends on the amount of the sample and how it was found. How old is the sample and how degraded is it? Restriction, fragment length, polymorphism, polymerase chain reaction, short tandem repeats, mitochondrial DNA analysis, and electrophoresis are all methods used to analyzing DNA.
We'll be on this unit for a couple of weeks, so as I mentioned, we're going to have a paper exercise today, and then we'll have some labs next week. Then we'll wrap this unit up probably in 2 to 3 weeks. Give you time to get these acronyms down. You'll have to keep track of them. They're not as difficult as they seem. RFLP, variations of lengths of the DNA. That's what an RFLP is. The PCR, that is a technique to, like a photocopier, make a whole bunch of copies, billions of copies of DNA so that you can work with it. And not only can you do DNA analysis off of the nucleus here in purple, so there's a lot of a lot of DNA in this nucleus, but what other organelle has DNA in it? Anybody remember from biology class? Correct. The things that are pink right here, they have their own DNA. And so that's what this mtDNA is. It's more difficult to get the DNA out of this, so they're only going to do it when the nucleus isn't there. So sometimes a degraded sample will have mitochondrial DNA still, where the nuclear DNA is damaged. Interesting thing about the mitochondria, have you realized that it's bacteria that they believe got engulfed by a eukaryotic cell? And symbiosis occurs because the mitochondria make energy, and the cell found out that it's got a great energy producer with the mitochondria in it, and the mitochondria get a nice place to live. So that's what symbiosis is. So early on in the development of cells, this self-surviving mitochondria, it's a bacteria, got engulfed, and now it finds itself in there. They divide on their own without instructions from the nucleus. And electrophoresis is taking electricity and separating DNA out. DNA is negatively charged, so if you put it into a machine like this, here's the negative terminal and the positive terminal. Whoops, took my eye out. And like charges repel, right? So if we put DNA in here and turn it on, this negative is going to push the negative DNA that way. This positive is going to attract it. And so the DNA gets separated out by size. So this is what it looks like. It's a banding pattern. And you know that genes come in pairs? We have 23 pairs of chromosome, one from mom and one from dad. Remember homozygous dominant, homozygous recessive? So they're showing capital letter A, it's a dominant gene, lowercase letter a. So if you are heterozygous, you have both forms of the gene. So it would show up as two different bands. If you're homozygous recessive, it's going to show up as one band. Homozygous dominant, show up as another band. And so this is where you get the variation. This is how they were able to determine the BTK killer. Remember that from our last video? They could tell who you're related to because you have gotten the gene from mom or dad. And so you would share some of the banding pattern together. All right, where did we leave off? Who do you want to read next?
Jessica.
RFLP.
Restriction fragment length polymorphism is a technique that shows variations from homologous DNA sequences that in turn individualizes it. The DNA sample is broken into pieces and is further assimilated by restriction enzymes, and then the DNA fragments are separated according to their lengths by gel electrophoresis. RFLP analysis was the first DNA profiling technique that was created by Sir Alec Jeffreys as a method of identifying individuals from their DNA. In 1986, Jeffreys used this method to help the authorities identify Colin Pitchfork as the murderer of two young girls in the English Midlands. Although RFLP was a breakthrough in the field of DNA fingerprinting, it is not ideal in forensics because it requires large amounts of DNA samples, which is not common in most crime scenes where small degraded samples are the reality.
So this is the first type of DNA that was used in DNA fingerprinting. It's a pretty big piece of DNA. So the larger the DNA is, the more that it can fracture, and so it wasn't as stable, and you needed a lot of it. You needed a visible pool of blood in order to do this. So up next is a process called PCR. When our cells are ready to divide because they get too large and inefficient, if this is going to split into two cells, you have to have twice the amount of DNA. So they figured out how DNA is copied in a regular cell. It's with an enzyme called polymerase chain reaction. So this here is not just a lab technique, it's what cells do in order to make a copy of themselves. So they figured out how it works. 3 steps. With temperature, it's going to make DNA open up because this is how DNA copies itself. My biology classes just did this last week and they had to make a movie of themselves in stop motion and explain the whole process. So it opens up so that each side can make a copy of itself. So they have a high temperature and it's going to just make this open up. And then if you have the right ingredients in there, the enzymes, and then all the building blocks of nucleotides, it will start to anneal, or it just makes another copy of itself, just all by itself. These chemicals come together. And then it will go all the way. So these are the individual pieces right here with enzymes that help it all along the way. So they figured out how cells do it, and then they put it into a machine, and they will raise the temperature, cool it down. They'll do that about a dozen times, and they end up with 1 billion copies of DNA when they start with 1. So now, in order to do DNA analysis, you just need 1 cell. If they can carefully extract the DNA out of that, then they can make copies of it. So that's what that polymerase chain reaction paragraph says. And usually it's going to be from skin cells because those are left behind very readily. You guys have already left thousands of skin cells at your table, and they just come over with a small little clean type of a vacuum, and then they have it in a little teeny bag, bring it to the lab, and start pulling the DNA out of it. So remember 13 was the number that I mentioned? Here they are, 13 core locations on our 46 chromosomes. So we have 23 pairs of chromosomes. The 23rd pair is the X and the Y. So they're showing the locations of where these 13 forensic pieces of DNA that they use. And let's do one more paragraph on short tandem repeats. Jessica, who do you want to read next?
The polymerase chain reaction is used to amplify a single copy or 3 copies of a piece of DNA, generating thousands to millions of copies of that particular DNA sequence. This enables analysis of DNA samples even from very small amounts of starting material. Like examining the DNA from cheek cell and saliva found on a glass, PCR is prone to error. Therefore, technicians must be very careful about the contamination, which can cause mutations in the DNA fragments.
Even identical twins will not be identical in all 13 of these locations because these aren't human genes in the sense that it's going to make us human, like with fingers and lungs. These are regions that don't code for anything. They are nonsense regions of the DNA, and we have a lot of variation in humans in these areas. And we'll talk about mitochondrial DNA a little bit later, but that's the last paragraph right there. And we are going to talk about the process. So we're going to do this now for on paper. And we've got a couple exercises with it, so you'll get it all down. Let's go over to our lab. I have it over here in Notability. Not yet. Who ate the cheese? Open that up. And there's a short video right here on gel electrophoresis. We'll take a look at that later. I wanted to go through the textbook of information because that's really useful for you to answer some questions in the lab write-up. So we used the textbook chapter just now, and that will help you answer the questions. Who ate the cheese? Just like its name implies, it involves cheese. Over in England, the Queen is having a ball, and she received a very expensive gift from a diplomat. So it was all set up for the banquet, and then someone came in and ate some of the cheese. So they're trying to find out who did that. They just took a bite out of it. They didn't use a fork, so their saliva is on the cheese. So that's paraphrasing the whole incident, and they did a really nice job of giving DNA type names to the suspects. So the first one is Princess Double Helix, or double helix, right? So that's what you call DNA in a double spiral. The spiral is a helix, and it's double because it has actually two sides to it. And you learned that last week. And you know the three parts of a nucleotide: a sugar, a phosphate, and a base. And they're held together in the center here with hydrogen bonds. There are 4 letters on the bases: A, T, C, and G. So that shouldn't sound unfamiliar to you. You'll be tested on knowing the parts of a nucleotide. So that's the whole process here of DNA analysis. So the princess was seen entering the sitting room, so she's well known for liking cheese. Suspect number 2, electrophoresis. Hey, that's what you call this process right here of a machine. You put a gel in there and you hook it up to electricity, and it separates the DNA out by size. So that is called electrophoresis, and they used her name. She is involved with a relationship with a Manchurian diplomat, and their relationship ended badly. So the diplomat gave the cheese, so maybe she is trying to make it look bad, ruining the party. Maybe she sabotaged the gift. Suspect 3, adenine. Hey, that's one of the 4 bases. Adenine is the letter A. Cytosine, guanine, adenine, and thymine are the 4 DNA bases. So adenine, she's the maid in charge of cleaning the sitting room. Well, she had access to the cheese. Maybe she took a little sample. Suspect number 4, Gene Tix, or genetics. Gene's the leader of the local cheesemakers guild. He probably doesn't like that his cheese wasn't at the party, so maybe he hijacked it. The crime lab investigator is R. Renee, so like RNA instead of DNA. So they took the cheese to the crime lab, and we have the DNA samples of our individuals. So we're going to do gel electrophoresis on 5 samples: the crime scene and 4 suspects. Sometimes this is done on paper, but I find that it's just as good using Notability here, so you don't have to cut things up with scissors. So I'm going to kind of go through the process with you. So you go through 1 through 5. After you're all done, we're going to compare the crime scene DNA to the suspects. So if you're taking scissors, you would cut this out, and it's got the name over each suspect so you don't lose track of it. If they were just all the letters, then you might go, which one is which? So this way you kind of keep it straight. So notice the red lines going up here. There's an enzyme that will cut every time that it finds two bases of C's and G's together. It always cuts right between the C's and G's. And so the first one has been partially done for you. So let's take a moment to find the C's and G's. Need to send this over to Notability. And then there's one more over here at the end. Oops. So now if this has been cut with the enzyme, then it's in a test tube, and you would take your pipette and you would get a sample of this and put it down into the well. Not knowing that it has 5 different pieces, right? 1, 2, 3, 4 different pieces. One is 7 bases long. This one is 4 bases long, and you're going to count up how many bases these are. Just by 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. So you put a 17 up there. That one's 4. And how many in the big one? 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 25. So here is your well. You're going to take, in real life, you would put your pipette down in that hole. That's a depression, a 3-dimensional depression inside of a gel, which is like Jell-O. You have to be careful not to poke through to the bottom of the gel. If you do, then your sample is going to go underneath all the gel. You want it to be inside the gel. So when you turn the electricity on, the negative terminal up here is going to push the DNA. So if you want to make a note for yourself and say DNA is negative, and then the positive pole down here is going to attract, so the DNA is going to be coming down, and it's going to get pushed down and pulled down as it reaches the other side. Notice the numbers along the side. This is 1 base pair, 2 base pairs. See, there's 7 from our crime scene. There was another one that was 4, right? So now you're going to take and make this four. Then we had 25. And was it 17? Yep. And 17. Oops. So there's our crime scene. That's the DNA off the saliva on the cheese. And now we're going to do that to the other suspects. And then there are a few of the questions for you to answer right there. So I'll leave the rest up to you. And this is due tomorrow, but it's a pretty quick thing. I think you can complete it on your own.